Afternoon. I apologize for the technical delays. However, I'm welcoming back to the second collision and the globe track, which is focusing on medical technology sector. To moderate this session, it gives me a great pleasure to introduce Manuela Cirilli. Manuela joined CERN Knowledge Transfer Group in 2010 after having studied both physics and science communication. With the KG Group, Manuela brings her unique blend of competences in the technical domain, medical applications, and communications. She is a deputy group leader since 2017 and currently leads the medical application strategy support team and the communication and training section. Thank you, Manuela.
Thank you so much, Anelia. I'm delighted to introduce our speaker for this session now, Dr. Martin McKeown. Martin is the director of the International Center of Excellence for Parkinson's Disease. This is the Pacific Parkinson's Research Center at the University of British Columbia in Vancouver, Canada. And he's also professor of medicine and adjunct professor in electrical and computing engineering at the same university. So Martin is a clinician scientist and he sees patients with mostly Parkinson's disease, and he also conducts research to help people with Parkinson's. He came to CERN during his undergraduate work in engineering physics, so it's a very curious trajectory, and we are all very curious to hear about his exciting research and also how training at CERN has been helpful to shape his career. So the floor is yours, Martin.
Okay, thank you. Unfortunately, I do have a few technical difficulties. I can't see any of the slides. I just see my own reflection.
We still have technical difficulties.
Okay, thank you. I'm not sure I can advance the slides. Can someone advance the slides for me, or? Okay, thank you. I will just assume someone can advance the slides for me. So thank you very much. I'd like to thank the coordinators of this event for inviting me. It's a great pleasure to come and talk to people. As Manuela mentioned, I'm a neurologist, a physician who deals with people with brain diseases. I mostly see people with Parkinson's disease, and we're involved in a number of projects. But I thought I would focus on two where I think my prior physics background helped me in these particular areas. And those are looking at novel treatments for Parkinson's and also disease monitoring. So if we could go to the next slide, please. So what is Parkinson's disease? Well, it's a neurodegenerative condition. And what this means is that there are certain cells in the brain that die prematurely. And this is probably, it's poorly understood, but it's probably a complex interplay between genetic and environmental factors. And the most prominent cells that die in the brain prematurely in Parkinson's are cells that secrete the brain chemical dopamine. So many of our treatments are related to replacing this missing chemical. Parkinson's is the second most common neurodegenerative disorder after Alzheimer's disease, and it affects up to 2% of people over age 65, and if you get into your 80s, it can be as high as 4%. It affects all races, and so it really is a worldwide phenomena. It seems to be increasing in prevalence, although we don't know whether or not we're just recognizing the symptoms more. But it certainly seems to be more prominent now than it was several years ago. Next slide, please. So about 50 years ago now, as I mentioned, because one of the most prominent neuropathological changes are the loss of these cells that secrete dopamine, it was found that replacing this brain chemical dopamine through medication was an extremely effective therapy. And so about 50 years ago was the introduction of a medication called levodopa, which is still the primary medicine that we use to treat this disorder. That was expected. What was unexpected was about 30 years ago when it was discovered that not only treating abnormal brain chemistry, but treating abnormal brain oscillations is also a very effective and proven therapy. And fortunately, who knows a lot about oscillations? Well, physicists are very good about this. And so this is why I think the physics community has a role to play in looking at these new therapies. So next slide, please. So this gives you some idea of whether or not we are asymptotically approaching the limit of what we can effectively do with medications. So the pharmaceutical industry, as you know, is a multi-billion euro industry, and they have very strong financial incentives to develop new medications. I just looked at Alzheimer's because it's even more prevalent than Parkinson's, and so there's even more financial incentive to develop pharmacotherapy. And you could see here that recently they tried to put through a drug, and it would have been the first drug in 20 years to slow the progression of cognitive impairment in Alzheimer's, and it was unsuccessful. And as I mentioned, for Parkinson's, we're still using the same medication that was introduced half a century ago. So this tells us that we need to be looking at alternate approaches for therapy. So next slide, please. So this has prompted people to look at techniques of brain stimulation. So on the left there is so-called deep brain stimulation, which is discovered about 30 years ago, and it is now used worldwide as an effective treatment. However, it's open brain surgery. It requires you to drill holes in the skull, insert electrodes deep inside the brain, and then have an implantable stimulator and a battery pack placed under the skin that sometimes has to be replaced maybe every 5 years or so. So this obviously, anytime you insert a foreign body into the brain, there's risks associated with bleeding and infection and so on. So there's a lot of interest in non-invasive stimulation, which you can see on the right-hand side of the screen. And it is possible to activate the brain by either applying focal ultrasound, applying powerful magnetic fields in transcranial magnetic stimulation, or applying electrical stimulation, either with a complicated waveform or stimulating behind the ear, which stimulates the vestibular system or the inner ear. Our lab has focused on the left side, which are these potentially portable areas because they require small stimulators that people could carry around, for example. So if we could go to the next slide, please. So our lab has focused on stimulating the vestibular system. So the vestibular system is a very curious system. It's a very old system that's seen in very low animals, and it's related to knowing where your brain and the rest of your body is in space. And it seems to influence very wide areas of the brain. And it's used a lot for visual stability. So when you're walking, the reason why the video frame doesn't appear jerky when you're walking is because of the vestibular system. It's important for balance. It's important for knowing where you are in space. And it's important for controlling things like the blood pressure. So, for example, when you stand up, the blood rushes to your legs, and so there has to be a reflex that maintains the blood pressure so you don't feel dizzy and enough blood gets back to your brain. And how does the body know that you've stood up? Well, it's the vestibular system. And so it seems to be a very ubiquitous system that's used for many different things. A few years ago now, one of the things that we discovered in our lab was that if we stimulate the system electrically by just putting electrodes behind the ear and give a very low stimulus, less than a milliamp, uh, and at a level so low that people can't even feel it, that we were able to manipulate brain rhythms, and including the brain rhythms that seem to be abnormal in Parkinson's. So this excited us because it seemed to be a non-invasive way where people don't even feel it, where we can manipulate the brain oscillations. And so we think that this would be a potentially complementary therapy to medication. We don't think it will replace it, but we do think that there are certain aspects where the medicine is not working sufficiently well, where we may want to complement it with this neuromodulation therapy. So next slide, please. So this is recent work from my former PhD student, Soo-Jin Lee. And what she did is she looked at different types of waveform stimuli. So as I said, physicists are very good at oscillations and looking at different waveforms that we could use. So what she did is we looked at a simple motor task where we measured reaction time. And what we did is we started off with a standard stimulus, which is a noisy or stochastic stimulus. And this is what is being used by our lab and other labs around the world as a potential treatment for Parkinson's. And we used that as a baseline. And what we did is we tried many other different stimuli. There were multi-sign stimuli. There was a simple linear superposition of sinusoidal waveforms in across a finite bandwidth. And what we did is we looked at the effects of improvements in reaction time compared to the baseline, which is the stochastic stimulus that's normally used. And what she found is that many of these other band-limited multi-sign stimuli were better as far as improving reaction time. But the key point was the optimum stimulus was a function of the individual. And we think that part of the reason why these technologies have not been more successful is they seem, if you use a one-size-fits-all stimulus, it works somewhat well for everyone. But what you need to do is choose the optimum stimuli for that individual, which has the capacities to significantly improve the benefits. And in medicine, we call this precision medicine, which is kind of an odd term, but it really means customized. So what we want to do is customize the stimulus to the individual. And this is why we are getting into data-driven approaches for this. So next slide, please. So this is one approach that we're doing. Now, if we want to test what is the best stimulus to, say, improve reaction time, obviously it's impractical. We have an infinite parameter space. We can't just have someone try millions and millions of trials of a motor task to check reaction time and check every different stimulus. So we have to be a little more clever about that. So one of the things that we're doing is we're using some brain monitoring technologies, including the electroencephalogram or EEG, which has got excellent temporal resolution, and functional MRI. Now, all of you are familiar with MRI scans. And if you think of an MRI scan as taking a picture of the brain, then a functional MRI are taking movies of the brain. And so we are using both the EEG and the fMRI to come up with signatures or high dimensional patterns which can predict what we are trying to target. So, for example, we would set up a model, a statistical model, and say, can we predict what are the brain waves that are associated with moving quickly, or what is the fMRI pattern associated with moving quickly? Okay, next slide, please. So once we have these signatures in the EEG and fMRI of, say, moving quickly, then what we do is we give a subset, a tractable number of calibration stimuli. And then we set up a different statistical model to say what what is the effects on our previously derived biomarkers or signatures in the EEG and fMRI of these standard stimuli. Next slide, please. And then if we can combine these together, what we can do then is have the computer go through and generate millions and millions of potential stimuli and use that to predict what would be the result on our EEG and fMRI responses, and then what would be the result in the reaction time or whatever we're trying to optimize, such as moving fast. And this will, we believe, enable us to come up with an optimum or customized stimulus for that individual. Once we have done that, then we can transport or offload that optimum stimulus to a portable stimulator, which is step 4 you see on the slide. And then step 5, what we want to do is then test the hypothesis that these customized stimuli will be substantially better than the standard stochastic stimulus that's normally used. So next slide, please. So once we've already built a prototype, these are fairly simple devices, but basically they are constant current stimuli, which means they're not really constant, but what we mean is relatively invariant to the impedance. And although with the current prototype we have fits on a belt, later, of course, when we optimize this, we can put it into a different form factor. So the ultimate idea would be someone wears a hat or something with this stimulator with the electrodes behind the ear and with it uploaded to their particular customized stimulus. So that's the sort of thing that we're envisioning. Okay, next slide, please. So the next thing that I would like to talk about is monitoring people with Parkinson's. So one of the things that's a bit counterintuitive is people's motor performance can actually fluctuate hour to hour. And people can go from actually having excessive movement if they have a little bit too much medicine to not enough movement if they don't have enough medicine. And that window, which is called the therapeutic window, can unfortunately be quite narrow for some people. So some people have a very difficult time where they can oscillate between having too little and too much medication. And when we see people in our clinic, we may see them as infrequently as every 6 months or every year. And from that short visit, we're trying to extrapolate how they're doing throughout the year. And obviously that's very difficult to do. So there's been a lot of interest in monitoring people with wearable sensors. The problem is that after a while, it's hard to convince people to wire up and put sensors on every day. And so compliance is quite low. So there's a lot of interest in using video monitoring. But of course, a lot of people have some privacy concerns. And so we've teamed up with an ethicist, and we've had various focus groups with the Parkinson's community about what sort of video monitoring they would feel comfortable with. We think we found a solution in that what we do is we developed what's called privacy compliant monitoring. And I'll tell you a little bit more about that in the next slide. But I just want to point out that in addition to longitudinal monitoring, automatically assessing people has also some benefits. And this varies from country to country. In my country, in Canada, we have many sparsely populated remote communities. And so we want to be able to remotely assess people. In other countries, they have too few Parkinson's neurologists. And so they don't have necessarily the expertise to be able to come up with an assessment of the motor performance. And so if you could automate this process, that would be helpful. And finally, if you had a new treatment and you wanted to monitor people, tracking the disease automatically would be very helpful. Next slide, please. So here's an example of what we call privacy compliant. We need the high resolution camera to be able to automatically recognize the correct person. But in real time, what we can do is we can convert that person into a stick figure. And this is the only thing that's saved. So if anyone were to hack into the camera, all they would see would be the stick figure. And I think a lot of people are much more comfortable with this. So as I said, their images are never saved and never transmitted externally. They are just in real time with so-called edge computing converted into these stick figures. And this still gives us enough information to monitor the disease, yet preserves the privacy of the individual. So this is something that we're working on. Okay, next slide, please. So here's an example. What we've done is we've used commercial sensors. There are many companies now that are marketing these sensors, and we're using this as sort of a label or a gold standard. And we're also doing the video. And ultimately, what we want to do is we're using the sensors to train a deep learning or machine learning algorithm so that we can use the video to ultimately replace the sensors. So we would get the same positional information that we would get from the sensors, but solely from the video. Next slide, please. So here's an example of this in real time. You can see it's not perfectly registered. We still have a few bugs to iron out, but you can get the idea that we can digitally zoom automatically into someone's hands, and we can look at the key points and then analyze these key points for mobility issues. And so this is the type of thing that we're doing. Next slide, please. And then finally, we're also looking at facial expressions. So people with Parkinson's have what's called a masked face. And so they have difficulty recognizing facial expressions, and also they have reduced facial expressions themselves. And what's kind of a fascinating aspect of the neuroscience of this is those two may be related, that in order to recognize someone's facial expression, you yourself have to have the ability to create certain facial expressions. And so we know that this is a problem in Parkinson's and in other diseases like Alzheimer's, people who may have difficulty with language. And so they may not be able to articulate that they are feeling anxious or unwell. And so we think some of these technologies will be helpful with facial assessment, especially if we have high resolution cameras that can automatically digitally zoom in and perhaps send an alert to a family member saying, grandma isn't feeling well or something like that. So these are the types of technologies that we're looking at. Okay, next slide, please. So that's all I had. I'm very happy to answer any questions. There's my email. So if anyone would like to send me an email later, I'd be happy to talk with them. So thank you very much. And I don't know, Manuela, how we moderate questions. Oh, there she is. Okay, great.
There I am. Thank you, Martin. That was really fascinating. So I think we have a little bit of a shy audience at the moment because I don't see questions coming through. So I'll profit to satisfy some of my own curiosities in the meantime. So I heard a story that your work on brain imaging was presented at CIA headquarters once. Can you tell us a little bit about that, or is it classified? Or
Yeah, so this is I think a bit of a funny story, but I was in San Diego working with a neural net pioneer, Terry Sinofsky, at the Salk Institute for Biological Studies, and we were looking at brain imaging technologies and brain imaging analysis methods. And when fMRI first came out, what you had to do is you had to hypothesize what the waveform would look like, and then search around in the brain for those parts of the brain that had that hypothesized waveform. So for example, if you had a task where someone was tapping their fingers and then resting, you would say, find me the voxels where there's activity and then rest and activity and rest. Well, the problem is what happens if you didn't know a priori what that waveform should be? In other words, if you didn't have a hypothesis-driven approach, how would you do a data-driven approach? And this is when we were the first to apply a technique called independent component analysis, or ICA, to fMRI. And this was a very nice data-driven approach to analyzing fMRI, which is now adopted throughout the world. What was interesting, though, is that the CIA contacted my supervisor and said, do you think this could be used as a fancy lie detector? So, of course, I'm a Canadian citizen, so they wouldn't ask me. So they asked my boss to fly up to CIA and present this new method for analyzing brain activity to see whether or not this could be a fancy lie detector. So that's the story of my slides being shown at CIA headquarters. I have no idea whether or not they used it or whether or not it was useful. I don't think it would be very useful as a lie detector, but.
Very curious to hear about it. Meantime, if we have a couple of questions on the vestibular waveforms and how often does a patient need the stimuli, every day, week, month, or are you still studying that part?
Well, that's a very good question. So it's interesting when you look at brain stimulus or stimulation. There's some stimuli that you give, and then you hope that there's a carryover effect. So, for example, transcranial magnetic stimulation, they use a very powerful magnet, but it's a huge machine, and because there's huge capacitor banks to generate the electric field or the magnetic field. So what you have to do is you have to stimulate people in the morning and hope that there's enough carryover effect to last for the rest of the day. Um, we haven't gone that route. We've actually gone with something that would be potentially portable. So this would be something that you would stimulate them all the time. We think that that effect is going to be much more stronger. There might be second order effects, but we haven't looked at that yet. We've mostly been focusing on the direct stimulation effect. So.
So I guess this is related to another couple of questions. One is that whether the patient needs to wear the device all the time, and a question that is about the cost of the technology, because it sounds amazing, but also very expensive, and yeah, especially the part about the customized brain stimulation. So maybe a comment on the outlook of it and how which parts and which progress would help in making it also an affordable technology.
Yeah, well, I think it's premature about knowing the cost. Actually, the stimulators, of course, are very inexpensive. They're battery powered. You're stimulating a milliamp of current. I mean, it's not a big deal. The stimulation, you're right. We are using expensive technologies to customize the stimuli. But we're not at the stage. What we don't know is whether or not you could have maybe a repertoire of 100 stimuli, and all you would have to choose is which of the 100 is best for that person. We don't know that yet. So we're very much at the research stage where we want to find the absolute best stimuli, and that might give us an upper bound of what we can expect from the technology. And then what we may find is if that's too expensive, as I said, maybe have 100 stimuli and say, okay, choose which of the 100 is best for that person, and that may be 80% is good. We just don't know yet, but that's I could see that might be the way forward ultimately. So,
thank you, Martin. An interesting question is also whether the video monitoring could also help chart the progression of Parkinson's.
yes, absolutely, and I think this is going to be. There's a lot of things that are where I think the benefit of this type of technology will be to find with data mining unexpected associations. So, for example, in Alzheimer's, if someone gets agitated, they may pace around the room more. And that's not something that you would be taught in medical school. So I think, it's finding these unexpected associations with certain movement patterns that might predict some aspects of the disease. Or, I mean, one of the challenges of Parkinson's is that it's a very heterogeneous disorder. And so it might be a way to stratify people and might be a way to predict whether or not some subgroup of people will respond to one therapy and not another. I think we're just starting to get into this, but I think we just need lots and lots of data to start doing the data mining. And I think some of these associations will start to fall out.
There's a question from the public which actually sort of ties with a question that I had myself. So the question is, how come the people who are diagnosed with this type of disease can remember very old times and not recent time? How this is really related with the chemical reaction in the brain? You mentioned at some point also Alzheimer when we were talking about facial expressions expressing feelings, but I guess also, for example, multiple sclerosis or other diseases. I mean, there's similarities across all these, so I guess integrated research could help and be beneficial to more than just one disease.
Yeah, well, I mean, we in clinical medicine, we often talk about whether or not people are lumpers or splitters. In other words, whether or not you try and look for commonalities across many diseases, or you try and find things that discriminate between the different diseases. And it is true that there are certain commonalities across the diseases, but there are also some very specific things. And I think your question was about dementia. Is that right? About recalling things remotely, but not. Yeah, that's very
Exactly.
fascinating. And basically what happens is that there's a part of the brain that encodes new memories. And so there's a whole area of what's called memory consolidation, where something happens, it's in a transient buffer, and then it basically gets translated into something that's much more permanent. And if you damage the area that's encoding the new memories, then, which is what happens in Alzheimer's and other types of dementia, then you have the consolidated memories because they were previously consolidated, but you lack the ability to encode new memories. And so that's why it's recent memory that's quite characteristically affected in Alzheimer's and similar diseases.
It's fascinating. I mean, just like when you mentioned that there seems to be a connection between the ability to have facial expressions and the ability to recognize the facial expressions. So, I mean, these are areas in the functioning of the brain that are really, really fascinating. I had questions because you mentioned several times, of course, functional MRI as well. Are you happy from the point of view of your research with the resolution of the current MRI machines? Is there something that you would wish to have to guide you better in the understanding of the brain and the diseases? It's a physicist question, of course.
Yes. Well, of course, more resolution is better, but the problem is with fMRI, you're actually measuring changes in the blood deoxygenated hemoglobin. So you're actually indirectly measuring blood flow. So the resolution may actually be on the physiological basis of that, not necessarily the physics of the machine. So I don't know if necessarily much stronger or much greater spatial resolution would necessarily help us. I think structurally it's very helpful.
Thank you, Martin. Thanks for answering the curiosities of the audience and also some of mine. And I guess it's time to move on to the next talk. So it's back to Anelia for the continuation of the session.
Okay.